Hello, and thank you for joining us. I'm Jasmin Nevarez, Digital Content Coordinator here at Simulations Plus. Director of Business Development, Dan O'Connor, will introduce our speaker for today in this webinar. Dr. Rodrigo Cristofoletti, Assistant Professor at the Center of Pharmacometrics and Systems Pharmacology at the University of Florida, will go over a case study leveraging IVIVE-PBPK modeling. Just a few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all the questions in the Q&A after the presentation. Go ahead, Dan.
Well, good morning, and thank you, Jasmine. It's my pleasure to welcome you all to our summer series of webinar as Simulation Plus continues to celebrate its 25th anniversary of institution, now coming into 26. But I was just speaking to our presenter this morning, and he's expecting 100-degree heat in Florida. I'm up here in Connecticut, not on the shore. This is imaginary, but we're expecting about 85, and Jasmine's in California, where I think it's going to be about 90. So it's definitely summertime, and we're looking forward to this presentation today that's on the subject of pharmacometrics. But before Rodrigo starts, just to give you a little bit of his background. Rodrigo received his PhD, summa cum laude, from the Johann Wolfgang Goethe University in Germany, under the supervision of Dr. Jennifer Dressman. I have no idea who that is, but I'm sure some of you do and know that that's reasonably important. He worked prior to joining the University of Florida. He worked in the clinical pharmacology office in the Brazilian Drug Regulatory Agency, Anvisa, where he was working to build the scientific foundation for regulatory policies within the Brazilian jurisdiction. Grew up in Brazil, went to Brasilia, and then recently, in the last couple of 3 years or so, joined the University of Florida. So without further ado, I'll turn it back to Jasmine, who will have a initial poll question. And then I believe, Rodrigo, it's up to you.
All right, so the first poll is, how long have you been using GastroPlus software? 0 years, 2 to 5 years, 5 to 10, or other. We just want to get a feel for what level of expertise we're presenting to. Give it a couple more seconds. All right, so it looks like most of you guys are beginners, and there are a couple 2 to 5 years, as well as 5 to 10. All right, we can go ahead and get started.
That's great. Thank you so much, Dan, for the very kind introduction. Jasmine, can you please just confirm if you are seeing my screen?
Yep, we see your screen.
So perfect. So thank you so much for the very kind invitation. So it's a pleasure to be here. And for today, my talk is going to be about using PBPK modeling to assess the impact of diseases on oral drug absorption. And I'm going to do that using a case study. So basically, I'm going to use a case study in HIV-infected patients. So this is just a very brief outline of the topics that I'd like to cover today. So we are going to start with a little bit of a background. So I will try to raise the research question that we are trying to tackle here. We are going to discuss about some of the key features in PBPK modeling that will enable us to tackle this research question. And I will use a case study focusing on ketoconazole to address all of that. So besides talking about using PBPK modeling to make predictions of drug exposure in special populations, I'm also going to use this study case here to talk a little bit more about translating in vitro biopharmaceutics data. So let's start from the beginning. So since 2012, regulatory authorities, they've been made significant efforts to enhance diversity of clinical trial populations. So we have important regulatory guidelines, they were published. So like the guideline for collection of race and ethnicity data. And we have this guideline here. This guideline represents a shift in the regulatory paradigm in the sense that the goal is not only selecting a rather homogeneous cohort of patients to reduce uncertainty around the estimated p-value, but the goal is to bring real-life variability to investigate the extrapolability of safety and efficacy profiles of a new drug across all target populations. Well, initiatives to enhance the diversity of clinical trial populations are not limited to the regulatory environment. We have significant efforts being made by patient organizations, academicians, and clinical pharmacology societies. So in this slide, we have a rather recent example from the American Society for Clinical Pharmacology and Therapeutics, where they are calling for new ideas. They are calling for articles focusing on harnessing diversity in disease biology and pharmacologic response to enable safe and effective use of therapeutic innovations in all patients. So it goes without saying here that studying clinical pharmacology in vulnerable populations, transgender population, minorities, all of that is essential to move towards health equity. So when it comes to enhance diversity of clinical trial populations, we have two complementary perspectives. On the one hand, we may focus on demographic characteristics like sex, race, ethnicity, and age. But we can also focus on non-demographic characteristics like organ dysfunction, comorbid conditions, and disabilities. Ideally, a study population should emulate demographic and non-demographic characteristics of real-life target population. We should include representatives from all patient subgroups. And this may be rather challenging, especially for drugs showing a wide prescribability potential. So for example, it's rather intuitive to think about enrolling HIV-infected patients on a clinical trial that was designed to assess safety and efficacy of a new antiretroviral drug. This is rather intuitive. But even though there's still no cure for HIV, people living with HIV are living longer and longer, and they are developing their own comorbid conditions. So they may get respiratory or enteric infections. And people living with HIV, they may also benefit from having access to a new antibiotic. They may eventually need a painkiller or an anti-inflammatory drug. However, it's rather rare to have the enrollment of HIV-infected patients in clinical trials that are not focusing on antiretroviral drugs development. There is this overall assumption that medically complex patients with comorbid conditions may be more susceptible to a potentially toxic effect of the investigational drug. So certain populations like people living with HIV are sometimes excluded from trials. And sometimes this exclusion is just based on an assumption, and we do not have a strong clinical or scientific justification. So despite all the efforts, enhancing diversity of clinical trial populations, especially for drugs showing a wide prescription potential, this is really rather challenging. And certain subgroups, especially related to chronic comorbid conditions, continue to be underrepresented in many clinical trials. In this context, using quantitative methods and modeling like PBPK modeling might be a very useful approach to assess the risks and help designing clinical trials. So for example, PBPK modeling has been widely used to assess the impact of organ impairment, liver and kidney impairment on drug exposure, and support subsequent design of clinical trials enrolling those patients. PBPK modeling can also be used for dosing optimization in special populations. So we hypothesized that we could use this PBPK-based rationale to gain mechanistic insights on the impact of comorbid conditions on the investigational drug exposure. And that may help us to better design clinical trials and to better decide about whether it's necessary or not to optimize dosing for those special populations. So in this talk, I'm going to present a case study using PBPK model to recapitulate drug exposure in HIV-infected patients. You've probably seen this figure before here. It's a very popular one among PBPK modelers. And this is important because it nicely illustrates the main feature of PBPK models, which is a complete separation between system and drug-specific parameters. As you know, PBPK model is a multi-compartmental model, attempting to represent individual organs and tissues in their physical arrangement, and they are bathed by the systemic circulation. Each compartment is defined a priori, and that definition is based on the knowledge about physiology and biochemistry for that specific tissue or organ. So most of the system-related parameters, they are derived from clinical data. On the other hand, drug-specific parameters, they are usually measured in vitro or animal models. So once a PBPK model is validated for a certain context of use, let's say explore PK in healthy adults, confidence is gained, and the model can be used to inform prospective simulations. So for example, we may investigate the impact of intrinsic factors like age, race, disease, or extrinsic factors like drug-drug interactions. We can impact those effects on drug pharmacokinetics. But since drug-specific parameters are usually measured in vitro or in animal models, the forward translation of these parameters within PBPK models is normally associated with uncertainties. Uncertainties related not only to the knowledge gaps in systems parameters, but also associated to the accuracy and translatability of the drug parameter itself. So in the absence of wide experience in forward translation of drug-specific parameters, the qualification of the overarching model can be obtained by verifying model performance through reverse translation. This involves fitting the model to observed data in optimizing drug parameters for which prior confidence is not too high. In other words, sometimes we need to move backwards and check if the model is able to reproduce already available clinical knowledge before moving forward to estimate still untested clinical scenarios, to make forward projections. And this is exactly the approach we have used in the case study I'm going to show you in the next slides. In a nutshell, we leveraged all available in vitro and in vivo data for the model drug in order to gain confidence in the translation of drug-specific parameters in healthy adults first. So it's a reverse translation approach where we are trying to gain confidence in the drug-specific parameters by reproducing PK studies in healthy adults. And once validated, this model can be used to make forward projections. So in this context, this PBPK model can be seen as a knowledge repository that can be exploited to gain insights related to a yet to be tested clinical scenario. So in other words, we are investigating whether a PBPK model developed and validated for ketoconazole in healthy adults, ketoconazole is going to be the model drug, whether this model would enable us to project the impact of HIV infection on ketoconazole absorption. So I gave you a spoiler. Ketoconazole is going to be the model drug. And ketoconazole is a classic representative of poorly soluble weak bases. It's a very old drug. It's rarely used in clinics, but there is plenty of clinical data available, including PK data in patients living with HIV. And this is essential for validating the model in special populations. So we are going to leverage all of that available clinical data. Also, ketoconazole is a textbook example. It has some very interesting PK properties. For example, besides showing a dose-dependent metabolism, it also shows a dose-dependent precipitation kinetics. And this was demonstrated by Dr. Cristofoletti's group. It was demonstrated in vivo when they followed the intraluminal concentrations of ketoconazole after administering different doses to healthy adults, and after collecting intestinal fluid and quantifying the drug directly in the intestinal fluid. So they realized that there is a kind of dose-dependent precipitation kinetics. So the secondary goal of this case study here is to investigate whether we could recapitulate this dose-dependent precipitation using PBPK modeling. Well, before jumping to the case study, let's briefly talk about model-based analysis of in vitro data. This is the concept behind this case study here. The concept is that by modeling in vitro data, we are able to derive fundamental parameters. So for example, by fitting a Michaelis-Menten model to in vitro data derived from liver microsomes, we may derive Km and Vmax values for a new compound. We may derive fundamental parameters describing the metabolism of that compound. By applying mechanistic compartmental uptake models to analyze data coming from drug transport across cell monolayers, we are able to derive important transport-mediated parameters. And the same rationale can be applied to biopharmaceutics data. However, here we have a plethora of in vitro biopharmaceutic systems. And consequently, we have multiple mechanistic models to consider. And sometimes it's a challenge to decide which model we are going to use to inform a subsequent PBPK model. But the bottom line here is that we can apply a model-based analysis to separate drug-specific parameters from in vitro systems parameters. So we have a deconvolution here. We have a deconvolution of in vitro data. We are moving from a process. The process can be dissolution or precipitation, and we are moving from that process towards parameters. Besides summarizing data, deriving those parameters is extremely useful for PBPK modeling, since those parameters, they are going to be inputs for the PBPK model. So in this study case, we will start by analyzing reported in vitro precipitation data for ketoconazole, data collected using dumping test, transmembrane flux, and biphasic dissolution. This analysis was done using the D+. So in all three systems here, we have ketoconazole fully dissolved in the donor or aqueous compartment. So as you know, the dumping test is a closed system to simulate precipitation due to pH shift. Since it's a closed system, it tends to overestimate precipitation rate, since there is no drug removal out of the system due to absorption. In the transmembrane flux, after complete dissolution of ketoconazole in gastric media, the donor chamber here was filled with simulated intestinal fluid. So drug supersaturation due to pH shift can be followed by drug precipitation in the donor compartment or by drug diffusion across the synthetic membrane into an acceptor vessel. And this is simulating absorption. So here we have an open system. So an open system combining dissolution and diffusion. So in D+, we can simultaneously model data coming from in vitro transmembrane flux experiments. We can model that data simultaneously for precipitation and diffusion. And this is the mathematical equation that we have in D+. I'm not going to get into the details for the sake of time here, but we can catch up later if you like. We have the same for the biphasic dissolution. Again, after complete dissolution of ketoconazole in gastric media, the vessel is filled with simulated intestinal fluid, and it's covered with an organic solvent, creating two phases. So drug supersaturation can be followed by precipitation in the aqueous phase, or drug can partition into the organic phase. Again, we have an open system here. So in D+, we can simultaneously model drug precipitation in the aqueous phase and also partitioning into the organic phase. And once more, this is the mathematical equation that we have in D+. So here we have the results. And for those results, we are modeling precipitation using a first order model, a first order decay function to describe drug precipitation. As expected, precipitation rate was faster when analyzing dumping test data. This is a closed system, so there is just one way for drugs in supersaturation. Drug will precipitate. And the mean precipitation time was estimated to be 350 seconds. Ketoconazole precipitation after pH shift was slightly slower in the transmembrane flux. It was 500 seconds here. However, due to the very small surface area of the membrane, this very small surface area, just 1.54 centimeters squared, it limits the diffusion of dissolved drug from the donor to the acceptor chamber. This is a limitation of this miniaturized system, and it really does not emulate the high intestinal surface area that we have in vivo for drug absorption. On the other hand, precipitation rate was almost 9-fold slower when estimated using data coming from the biphasic dissolution system, from the in vitro biphasic dissolution system. So here we hypothesized that the biphasic dissolution data would give us a better input for ketoconazole PBPK model. We would get a better translation of precipitation using data coming from the biphasic dissolution data, since we have a higher surface area getting closer to the highest absorptive surface area that we have in vivo. So right now, we have estimated parameters using D+. We are going to get those parameters, and they are going to be inputs for the GastroPlus. We are going to use GastroPlus to translate in vitro parameters. But before showing you the results, I'd like to get back to Jasmin, and she will have another question for you.
Thank you, Rodrigo. So our second poll is, what are your primary interests in joining us today? And then you can choose more than one answer. And I'll give everybody a couple seconds here. All right. And it looks like there's a good mix. Most of you chose PBPK modeling and then oral drug absorption. Back to you, Rodrigo.
Thank you very much, Jasmin. So let's see the results. So basically, right now, we are moving from D+ towards GastroPlus, and they go well hand in hand. You know, we are using D+ to analyze in vitro data to derive fundamental parameters. And we are going to use G+ to translate those parameters and estimate pharmacokinetics for ketoconazole. So right now we are applying this translation step. So right now the goal is to investigate which drug precipitation, which precipitation rate would enable us to recapitulate the clinical pharmacokinetic profiles obtained after administration of ketoconazole 200 milligrams as an oral solution to healthy adults under fasting conditions. So I think it's important right now to mention that since we do not have IV data for ketoconazole, we are applying a stepwise modeling workflow here. So for this simulation, we do have a mechanistic model describing absorption. We are using ACAT, but the distribution and elimination of ketoconazole is being described using a compartmental model that was parameterized by fitting a compartmental model to the PK data, and also using some liver and gut extraction data derived from clinical studies. So the goal here was really to focus first on the mechanistic absorption model. So in this plot here, you can see the results. So as you can see here, represented by the blue line, we have that the precipitation rate that was derived from the dumping test is really underestimating the pharmacokinetics of ketoconazole. The same is happening with the parameter derived using the transmembrane flux. So on the we can also say here that we are really overestimating the in vivo precipitation of ketoconazole. So the red symbols here, they represent in vivo results. And it seems that we can better capture the in vivo results when using a rather slow to almost no precipitation in the model. We expanded this modeling exercise towards additional doses, 400 and 800 mg, both administered as oral solutions to healthy adults under fasting conditions. Same drill here. Disposition models, they were fitted to the respective clinical data. So you can see a change in the apparent clearance here, since ketoconazole elimination is nonlinear. So on the left-hand side of this slide, we have the results for 400 milligrams. So again, the PK profile, the observed PK profile, would be better captured if we have used a precipitation rate between the value calculated from in vitro biphasic dissolution data, the green line here, and no precipitation at all. In turn, for the 800 milligrams, the PK profile was nicely captured when using the precipitation rate derived from in vitro biphasic dissolution data. So at this point, we are describing ketoconazole precipitation using a first-order decay function. And this function does not have enough granularity to account for differences in doses. In other words, we are using exactly the same precipitation time for all doses, for 200, 400, and 800 mg, which does not seem to be adequate for ketoconazole, since a precipitation rate in vivo seems to be dose-dependent. So in this context, we decided to move backwards to D+ and try to analyze in vitro precipitation data coming from the biphasic dissolution, which was the most promising one. But now, instead of using the first order model, we are using a mechanistic model. We are using the built-in link force model, which is a mechanistic model based on the classical nucleation and crystal growth theory. On the right-hand side here, we have the equation for the link force model. So basically, we have here diffusion coefficient, Avogadro's number, we have concentration. C stands for concentration. So in the link force model, it considers concentration as a variable affecting precipitation. So in D+, we can fit lambda, which is the effective radius. And also we can fit this empirical exponential correction factor. All other parameters, they are estimated automatically, including the critical radius, R, including the interfacial tension, gamma, and the molecular volume. So here, we fitted lambda and the exponential correction factor to the in vitro biphasic dissolution data. And here we have the plot showing observed symbols and simulated data, the lines. Blue symbols and blue line represent data in the aqueous compartment, and orange ones represent data in the organic compartment. So you can see there's a very good agreement between simulations and observations as well. So it was a fitting exercise. And here we have the fitted parameters. So you know the drill right now. We have derived fundamental parameters in D+. So right now we are going to use those parameters as inputs for the GastroPlus. And here we have the results for the 3 doses. Symbols represent observed data, line represents simulation. As you can see, unfortunately, the forward translation of link force parameters kind of failed. And we ended up underestimating ketoconazole exposure for all doses, from 200 mg to 800 mg. So we have the model now overestimating ketoconazole precipitation. So this is really telling us that we have uncertainties related to the translatability of precipitation data using the mechanistic model. And further research is needed. So it seems that simply by fitting lambda and the empirical exponential correction factor, we are not able to translate this precipitation in vivo. So it seems that we may have some uncertainties in the automatic estimation of the other parameters related to the link force model. So further research is needed here. And in order to gain confidence, we applied a reverse translation approach. This is always the drill. So every time that we have uncertainties around the forward translation of drug-specific parameters, we can gain confidence by applying a reverse translation approach. So here we simultaneously fitted the 2 link force parameters to PK data from all 3 doses. Now the model is able to capture the dose-dependent precipitation kinetics for ketoconazole. We can see a significant precipitation for 800 milligrams, a smaller degree of precipitation for 400 mg, basically no precipitation for 200 milligrams. So at this point, with just one set of precipitation parameters, link force parameters, we are able to explain ketoconazole precipitation after oral administration of 200, 400, and 800 milligrams of ketoconazole as oral solution. It's a progress, but at this point, we still do not have a full PBPK model. We have a unique ACAT model that is connected to 3 different compartmental models describing ketoconazole elimination after 200, 400, and 800 mg. Remember, it was a stepwise modeling workflow. So for the next step, we are going to replace the compartmental model by a full PBPK model. So right now, we are going to estimate tissue to plasma partition coefficients and volume of distribution using a mechanistic model. We are going to use the Lukachova method, and we are going to estimate Vmax and Km for CYP3A4. We are assuming that CYP3A4 is the only enzyme mediating ketoconazole metabolism. And we are going to estimate those 2 parameters by simultaneously fitting Vmax and Km to the PK data that is available for 200 to 800 milligrams of ketoconazole oral solution. So right now, the resulting model was able to capture the non-linear elimination of ketoconazole. As you can see here, a 4-fold increase in dose from 200 to 800 milligrams was followed by almost 9-fold increase in AUC. So we are moving from 18 to 161, almost 9-fold increase in AUC, characterizing the non-linear elimination of ketoconazole. So right now we have one ACAT-PBPK model able to recapitulate ketoconazole dose-dependent elimination and ketoconazole dose-dependent precipitation. So we are gaining confidence in drug-specific parameters. We are gaining confidence in the translatability of drug-specific parameters. So the next step was to expand the model to simulate systemic exposure after oral administration of ketoconazole tablets. We are moving from solutions to tablets. One way to do this is by simply inputting the in vitro dissolution profile in terms of fraction dissolved over time directly into GastroPlus. We can do that. It's very straightforward. However, the caveat is that with this approach, it assumes that the same dissolution profile will happen in the gastrointestinal tract of every virtual individual. So we are bypassing the gastrointestinal variability. Alternatively, we may model in vitro dissolution data. We may model in vitro dissolution data using a mechanistic model, like the Johnson model that is represented here in this slide. The Johnson model is a diffusion layer model. And this model-based analysis of in vitro dissolution data will enable us to deconvolute the in vitro dissolution data, to deconvolute the dissolution as a process, separating drug and in vitro system component. So we are moving from process to parameters, deriving fundamental parameters. In this case, we use the Johnson model to derive a product-specific particle size distribution, or PPSD for short. In other words, we are deriving a particle size distribution that, together with the diffusion coefficient of the drug, together with density, thickness of the diffusion layer, and solubility, would explain the in vitro dissolution profile obtained for ketoconazole tablets in FASGIVE. Obviously, this approach might be hindered by identifiability issues. So it's really important to investigate if this PPSD would really enable us to explain the product behavior in different experimental conditions. So we are talking here about running multiple predict, learn, confirm cycles using in vitro data. So in this case here, we were able to explain the ketoconazole tablets dissolution in FASGIVE, FASIF, and FASIF using exactly the same PPSD, mitigating concerns related to identifiability issues. So at the end of the day here, we are breaking in vitro dissolution down into its mechanistic components. We moved from the process, dissolution, to parameters. And right now, we are going to use those parameters as inputs for GastroPlus. So in the previous slide, there was the deconvolution of the in vitro dissolution data. So right now, we are going to reconvolute the input that we derived, the PPSD, but now we are going to reconvolute this parameter. We are going to move from the parameter back to the in vivo dissolution in vivo. So right now, we are going to integrate the drug and formulation parameters with gastrointestinal physiology to estimate in vivo dissolution for every virtual subject, taking gastrointestinal variability into account. So we are integrating drug-specific parameters with physiology. So we are running a deconvolution in vitro to estimate fundamental parameters, and we are reconvoluting that fundamental parameter in vivo. We are using the same mechanistic model, Johnson model in D+, and Johnson model in GastroPlus, to reconvolute, reassemble the dissolution, but now in vivo. So the resulting model was able to recapitulate ketoconazole pharmacokinetics after oral administration of 200, 400, 800 milligrams as tablets to healthy adults. So as you can see here, the ratios between simulated and observed PK metrics were within bioequivalence criteria, which is a very stringent acceptance criteria for modeling purposes. Same results were observed right now under fed state conditions. So briefly, food does not significantly affect ketoconazole Cmax or AUC. The major impact here is on Tmax, which is delayed due to the slow gastric emptying after a meal. And this Tmax delay was nicely captured by the model. So at this point, we have a model that is able to recapitulate the PK of ketoconazole when administered as oral solution or tablets. It's able to recapitulate ketoconazole dose-dependent elimination, ketoconazole dose-dependent precipitation kinetics, and food effect. So it's fair to conclude that now we have a reasonable level of confidence in the drug-specific components of the model. We have confidence in the translatability of in vitro parameters, translatability of drug-specific in vitro parameters. We gained confidence using the reverse translation approach. So at this point, we are ready to apply this model to simulate ketoconazole pharmacokinetics in still untested clinical scenarios. In our case, we are going to apply this model to simulate ketoconazole pharmacokinetics in people living with HIV. But in order to do that, we really need to understand what is the impact of HIV infection on physiology. There we go. Overall, we may stratify people living with HIV in two groups in terms of CD4 cell count. So we have those with CD4 cell count above 200 cells per microliter, representing patients who are adherent to their treatments, and thus they are patients less susceptible to opportunistic infections. And we may also have patients with CD4 cell count below 200 cells per microliter. And those patients, normally, they show deficient immune systems, and they are much more susceptible to secondary infections. As you can see here, gastric pH, doesn't matter the CD4 count, is significantly higher in people living with HIV in comparison to healthy adults. Unfortunately, the mechanisms behind it, they are still not fully understood. But as you can see here, enteric infection seems to be a critical variable. So after reviewing the pertinent literature, we estimated a mean gastric pH of approximately 3.5 for patients showing CD4 cell count above 200. And a gastric pH of approximately 4.9 for patients showing a CD4 cell count below 200. So we have that people living with HIV, their stomach, we have hypochlorhydric conditions in their stomach. Remember, we are talking about a weak base as a model drug. So we can really anticipate that this may cause, this may impact the absorption of ketoconazole here. So we have, this is just one of the impacts of HIV infection on the physiology. HIV infection also seems to affect gastric emptying time. So same drill. So this is dependent on the CD4 count and also dependent on whether there is a secondary infection or not. Overall, gastric emptying is threefold slower in HIV-infected patients with CD4 cell count below 200 cells per microliter. So we have two major changes in the gastrointestinal tract of people living with HIV. There is a change in gastric pH, we have hypochlorhydric conditions, and we have a change in gastric emptying time. There is a delay in gastric emptying time. So these changes, we are going to incorporate those changes in physiology in the PBPK model. And we are going to run simulations to project the impact of HIV infection on ketoconazole absorption. So basically, the drug-specific components of the model, the drug-specific components, they will be fixed. And we are going to replace the system components from healthy adult to HIV-infected patients. We are going to move from a healthy GI tract to a GI tract showing the impact of HIV infection. Here we have the results. So the plot on the left-hand side is showing ketoconazole pharmacokinetics in healthy adults. Line represents simulations, and the symbols represent clinical data. On the right-hand side, we have ketoconazole pharmacokinetics in HIV-infected patients with CD4 cell count below 200 cells per microliters. So we are talking about people living with HIV, but also with a very deficient immune system. So as you can see here, the model was able to capture the more than threefold decrease in systemic exposure of ketoconazole in HIV-infected patients. So the only difference between both models is in terms of the physiology. It's related to the systems parameters. The drug-specific parameters, they are exactly the same. So expanding this research towards other drugs for which we have clinical data in HIV-infected patients will certainly help improving confidence in the systems parameters that are altered in HIV-infected patients. And we really hope that in the near future, after applying the same conceptual rationale, this reverse translation approach, and leveraging available PK data for other drugs in people living with HIV, we will be really able to create a virtual model for people living with HIV. And hopefully that model is going to be available in the GastroPlus library to inform subsequent simulations. So once this model is developed, we can use it to gain mechanistic insights on the impact of HIV infection on ketoconazole absorption. And here we have a very interesting result coming out of a parameter sensitivity analysis. So basically, in this plot here, we are investigating what is the impact of increasing the particle size distribution and consequently decreasing the dissolution kinetics in terms of Cmax. So results showed here that the Cmax in HIV-infected patients, represented by the blue symbols, is much more sensitive to changes in particle size, to changes in dissolution, than the Cmax in healthy adults. So this is telling us that changing formulations might have a higher impact on ketoconazole exposure in HIV-infected patients in comparison to healthy adults. So this is really something that we can also try to make an interpretation from the bioequivalence perspective. So from the bioequivalence perspective, we run BE trials in healthy adults. This is the regulatory model. And we assume that that model is the most sensitive to identify differences in formulation if they exist. And then we can just extrapolate the results across all patients. And this, we are getting a different message. It seems that in people living with HIV, they should be more sensitive to changes in dissolution when it comes to formulations containing ketoconazole. So here we are applying the knowledge that we have been accumulating in the PBPK model. We are exploiting this knowledge repository. So this type of models will be rather useful to assess the risks of enrolling HIV patients in clinical trials. And this PBPK model will also be rather useful to inform dosing adjustments in that specific population. So just to wrap up, so we saw that model-based analysis of in vitro data is helpful to derive fundamental input parameters for PBPK models. even for biopharmaceutics data. Generalizing first-order precipitation rate across different doses is not straightforward. We should be aware that for some drugs like ketoconazole, we may have dose-dependent precipitation, and using first-order models to describe precipitation might not be a good idea. On the other hand, forward translation of in vitro precipitation data using linked force model still requires further research. We still have uncertainties related to the forward translation of in vitro precipitation data using linked force model. So gaining experience in translatability of drug-specific parameters is needed to inform forward projections. In this study case, we were able to recapitulate the impact of HIV infection on the ketoconazole oral absorption. Obviously, the critical part here is the knowledge in terms of the system parameters. It really requires a knowledge in terms of what is the impact of the disease in terms of physiology. So at the end of the day, PBPK modeling like this may be useful to gain mechanistic insights on the impact of comorbid conditions on the exposure of the investigational drug, and it may help us assessing the risks related to enroll those patients in clinical trials. And most importantly, PBPK modeling may help us to support subsequent dosing optimizations for special populations like people living with HIV. So just to finish my talk today, I'd like to thank my group. This is really definitely not one-person research. So I'd like to thank Yesenia Franco, Lai Silva, Brian Cicali, Nitin Charb, Hannah Kinvig. So this was a multi-hand work. And also I'd like to thank Simulations Plus for sponsoring this research. Hopefully by expanding this modeling exercise towards other drugs, we may have a virtual model for people living with HIV in GastroPlus, and this may help us to run subsequent simulations to get a better understanding of pharmacokinetics in that special population. So thank you so much. Now I will hand it back to Jasmin and Dan. And then I'll be more than happy to try to address any questions that you might have.
Okay, Jasmin, are you going to want me to take over, or how would you like to proceed?
Go ahead, Dan. You can pick out which questions.
Okay, great. Yeah, we do have a couple of questions for you, Rodrigo. Yeah, a great presentation, and always wonderful to be able to look at disease populations and be able to predict. I think that's incredibly important. We had a couple of questions around the surface area and transmembrane flux. I'll consolidate the two questions into one. Would it be better to look at surface area to volume ratio rather than just surface area alone? And will the correlation improve if increasing the transmembrane flux surface area? So I don't know if you can, I've conflated those, but you're welcome to take a shot.
I definitely think that's a very good point. So, I mean, definitely, I think we can use that approach to try to scale up a miniaturized system. I think that definitely would kind of mitigate the impact of the smaller surface area in the transmembrane system. I would say yes. I think that's something that we can consider, and that would probably improve the predictive capacity of that system. I think that's a very good point. Fully agree.
All right. We still have a little time for a couple other questions. In DDD plus, is the precipitation rate simulated, or are you calculating it from the experimental data?
Very good question as well, then. So we are calculating the precipitation kinetics first using a first order model, and after that using the linked force model. So we are calculating the precipitation kinetics using the in vitro data.
Right. And back and forth, it can be both directions, so to speak.
Exactly. Exactly.
Yeah. If you don't have human observation data, then animal models can give you better observation of in vitro dissolution. So in other words, if you don't have human data, can animal models give you better observation of in vitro dissolution?
Well, I think it's a very good, I mean, theoretical, I would say it's always important when you are trying to gain experience in terms of translating in vitro data. It's always important to have some data that you can compare to. So just to see how well or how far away the translation is kind of taking us. So I do not have much experience in terms of translating in vitro data and validating the translation using preclinical data. So, but I would say that it's important to have some in vivo data. This is really telling us whether we have confidence in using that or not. So I guess that one way to do this would be by replacing the human model in GastroPlus and by using an animal model there. So conceptually, I would say yes. We have never done that in the lab. So normally we are doing this translation exercise and we are translating from in vitro into humans. But I would say that conceptually you could do the same using an animal model as well.
Yeah, and only in rare occasions does rat model work for a human, you know, in those people that are not rats. But yeah, drug interaction. Did you consider it in this model where HIV patients under any active treatments?
That's an awesome question. Yes, we are considering some perpetrators there, like efavirenz, for example. There is an inducer of 3A4. So I think this is the beauty of this one, because you can really get a very broad understanding of what might be happening there. So let's say, well, let's say that we have an investigational drug that is also a C3A4 substrate. So let's say that that drug has some physical chemical properties quite similar to ketoconazole. So we are bringing exactly the same rationale here. So, and I think one of the questions in terms of including or not people living with HIV is that those patients, they are taking some medications, and some antiretrovirals, they really can affect C3A4, for example. And I think this is the beauty of this, because you can really run a very broad risk assessment there, and you can really try to understand what would be the impact of that co-medication if you are bringing those patients to the clinical trial. And yes, we are running right now, we are really exploring efavirenz impact on all of that. So, yep.
Right. All right, and we have a minute or two left. I'll try to get a couple more in. What strategies are available to assess the impact of the disease where you know that the disease impacts intestinal permeability, especially for oral administration, obviously, that are expected to have low intestinal absorption?
Well, I think you may have a couple of ways to deal with that. So just to give you, just to have a concrete case here. So we've been working with, for example, patients being affected by post-bariatric surgery. So you can just go there and try to modify the system's parameters themselves. So, but somehow, if you have an information that, if you have some data that, some experimental data, in vitro animal data that is really telling you that there is a decrease in terms of the permeability. So let's say that you have an in vitro model, we are trying to work with this. So we have this hypothesis, just not to give a concrete case. So we have this hypothesis that drug absorption in Down syndrome patients might be kind of impaired, since we have loss of down regulations due to the extra chromosome. So basically, we are trying to derive some in vitro systems representing the intestine of Down syndrome patients. We are working with organoids and gut chip technology. So there we are trying to derive this apparent permeability data, and that we can use to try to generate a path value. That could be a strategy. So you can try to generate in vitro data that would give you a quantitative idea what is the impairment of permeability that you may have with a certain disease. And then with that data, you can reparameterize your model as well. This is a way to go. So if you are talking about a downregulation of transporters, for example, and you have some proteomics in that special population, you can try to reparameterize your intestine, for example, kind of representing this low expression of some of the transporters. So I would say it depends a little bit on what the disease is causing in the intestine. So you can come up with different strategies. So you can go there and directly change the system component itself, or you can try to derive an in vitro system that will enable you to get a better estimate of the impact of the disease in terms of permeability and use that value in your model.
All right, great. And we've run up against the clock. I just close with, there have been a couple of questions about obtaining access to GastroPlus or DDD Plus. Both of those avenues are available on our website, or you can reach out to myself or Jasmin, and we'd be most happy to follow up. Jasmin, I'll throw it back to you to close.
All right, thank you, Dan. Thank you, Rodrigo. We invite our audience to learn more about GastroPlus or any of our other software, again, at our website, www.simulations-plus.com. You can also find us on LinkedIn, Twitter, and Facebook. This webinar has been recorded for playback and will be available on our website and YouTube channel. You can also find more information about the University Plus program or links to our socials in the chat. And any questions we may have not answered, we'll be answering on LinkedIn. This concludes our webinar for today. Thank you, everyone.
Thank you very much. It was a real pleasure to be here today. Thank you for having me.
Bye. Thanks, Rodrigo.
Bye.